Now we are going to be looking at the chemical properties and reactions for fats and oils. First, let's look at synthesis. Lipogenesis is the metabolic process of creating fat from non-fat sources, primarily excess carbohydrates and sometimes proteins or alcohol. Even though we are not going to look in detail at the metabolic process, understand that the synthesis of triglycerides basically consists of the fusion via an esterification reaction of specifically an alcohol molecule, that in this case we have glycerol as our representative of the alcohol. And then we are going to be putting together this alcohol with a carboxylic acid. And the carboxylic acid is going to be provided by the fatty acids that are going to be present in the triglyceride. And as you can see, I'm going to highlight the important functional group. Once the alcohols react with the carboxylic acid, what we have is an esterification reaction, which is mediated specifically by enzymes in the body. But overall, what you should observe and pay attention to is that the C double bond O from the carboxylic acid, in this case our fatty acid, is now going to be bonded to the oxygen in the glycerol. Understand that overall, we're going to form ester bonds. And specifically in a triglyceride, we have 1, 2. Let me include the carbon because it's more correct to include the carbon atom because that is the signature of the ester. One ester, 2 esters, 3 esters are present. Now, let's also look and take into consideration that the OH from the carboxylic acid came together with the hydrogen in glycerol, and that's where water comes from in the synthesis of a triglyceride. Triglycerides, because they have the ester bonds, they can undergo hydrolysis. We learned previously that esters can be hydrolyzed under acidic conditions. And when that happens, we are going to be forming an alcohol and a carboxylic acid. But if we start with a triglyceride, we need water and acid, or water and an enzyme, in this case, lipase, which is going to act as our catalyst to take the 3 ester bonds in the triglycerides into a single alcohol molecule, which is glycerol, along with the 3 different fatty acids that were present in the triglyceride. As I mentioned a few moments ago, you can see that in equation 14.2, we are going to have a mixed triglyceride. So triglycerides not always have the same fatty acid in all 3 positions. We can observe that in this equation, we actually have a triglyceride that has different fatty acids. But overall, understand that upon acid hydrolysis of a triglyceride, you will form glycerol and you will form specifically three fatty acids. We also learned previously when discussing organic chemistry that esters can be hydrolyzed specifically in the presence of strong base. That strong base, let's remind ourselves that is going to be aqueous. So when you take a triglyceride and you expose it to aqueous, a strong base, we are going to be doing a saponification reaction. In a saponification reaction, the product of the reaction is going to be an alcohol in the form of glycerol. And additionally, we're going to be making soap. But if we relate this to what we learned previously when we were looking at hydrolysis of esters, the glycerol, as I just mentioned, is our alcohol. And these soaps is just another form of a carboxylate salt. In a carboxylate salt, in the case of this type of hydrolysis of a triglyceride, is going to be the soap. Soaps act as cleaning agents. And as you can see in figure 14.3, the use of soaps are often used by health professionals because this is a way to control the spread of infectious diseases or infectious microorganisms in the case that a surgery has to be performed. One of the things that soap molecules can do is form micelles. Micelles are spherical clusters of molecules in which there is a polar portion that is actually at the surface of the micelle, and we have a non-polar portion that is located in the interior of the micelle. We can see that the structure of a micelle, specifically, it is illustrated in part C of figure 14.4. Overall, when we look at specifically part A and part B of figure 14.4, we can see that in part A, we have the molecular structure of a fatty acid. Specifically, this is showing us the structure of lauric acid. Now, let's remind ourselves that if we have a soap, basically this portion of the molecule doesn't look like a carboxylic acid. In a soap, that portion of the molecule looks at C double bond O connected to an O minus, and that O minus is paired with a cation. In this case, I'm choosing to illustrate it as sodium. Regardless, if you have a soap or if you have a carboxylic acid, having a carbon-oxygen bond represents a polar bond because of the electronegativity difference between the atoms. So regardless if you have a soap or if you have a fatty acid, this area of the molecule is going to be specifically the polar section of the molecule. On the other hand, because carbon-hydrogen bonds are going to be non-polar, we can see that that section of the molecule is known as a non-polar tail. When it comes to their interactions of these two sections of the molecule with water, so the interaction with water, we see that the polar side of these structures are going to be interacting with water, but then the non-polar tail is going to be water insoluble. In biochemistry, typically we don't write a lot of the functional groups. So understand that in letter B, all we're doing is using a cartoon to illustrate the structural data that we see on letter A. Yeah. Typically, a circle, it is utilized to represent the polar head, and then a squiggly line represents the non-polar tail. So when in letter C, we see specifically a micelle that is formed by carboxylate groups like we see in soap, and we see the nonpolar tails, we can see that in micelles, all these soap molecules are going to be arranged where the tails meet each other. And then the carboxylate side, understand that the sodium is not included, is going to be facing outside of the cell. Now, that C double bond O, which is represented here as COO minus, is going to be hydrophilic because those groups can interact with water. The nonpolar tails, since they are inside of the micelle, they're going to be representing hydrophobic groups. They will not like to interact with water, so that's why they are in the interior of a micelle. One of the things that is not illustrated here, but understand that it is important in soap action, is that when it comes to soap removing soiling agents that are nonpolar, because of the attractive forces that will be happening between things that soil surfaces. Let's use as an example oil. So let's say that you have a specific surface that has a piece of, let's say, and I'm going to represent it by this green blob. It has something that is soiling the surface, and it happens to be oil. When a soap acts on that surface to remove that oil, understand that this area of the molecule, the tails, are going to be interacting with that oil and overall trapping it in the interior of the micelle. And since the surface of the micelle can interact with water, this is how things are removed, because since soap molecules can make micelles, basically it's going to trap the oil in the middle, and then it is, in a way, since the micelle is water soluble because of how it's structured, that's how oils that are basically soiling surfaces can be removed. Continuing on with the reactions that can happen in triglycerides, now we have the process of hydrogenation. Hydrogenation is going to be a reaction that we have seen previously in chapters, including when we were hydrogenating alkenes, and we also saw that this is a reducing reagent. But regardless, in this instance, when we have specifically hydrogenation, the hydrogenation is going to be acting only specifically on the carbon-carbon double bonds in our molecules. So if you have an unsaturated triglyceride and you expose it to H2 and a catalyst, understand that what you're going to be synthesizing is going to be a more saturated triglyceride. And that is because upon reaction of the H2 and catalyst with the unsaturated triglyceride, we see that only a portion of the molecule will be receiving those hydrogens. And we see that that carbon-carbon double bond turns into carbon-carbon single bond, and we are adding hydrogens on the alkene. We are observing in this example only partial hydrogenation. During partial hydrogenation, it is important to note that because of the reaction itself, that's why we have food products that are like shortening, butters that are plant-based. They can be created by this reaction, but only partial hydrogenation is what is required. One of the issues with partial hydrogenation, and maybe in the food products that you may consume, you use products that says partially hydrogenated oils, then this is the reaction that is occurring. Mm. One of the problems with this reaction is that one of the side products of the reaction is going to be the conversion of cis fats into trans fat. These trans fat at times could be found in products like margarine, shortening, or even high-fat baked goods. But nowadays, you can also observe that in food products, you may see that in the label it already says it contains partially hydrogenated oils with no trans fat. That means that the company that is producing this food product actually removed the trans fat that were produced within the reaction. Nevertheless, understand that partial hydrogenation is needed in order to create these food products, because if you do full hydrogenation on the unsaturated triglyceride, then it's going to create a product that, to be honest with you, is not going to be stable. Please make sure that when you look at the homework, when it comes to this reaction, you observe if the question is asking for partial hydrogenation or full hydrogenation. Because if the question asks for full hydrogenation, then understand that every single double bond is going to be changed into single bonds. Here we can observe a reaction map specifically for triglycerides. At the top of this flowchart, we can see that the formation of triglycerides is produced from glycerol and fatty acids. These molecules are going to lose water in the process, and they will be able to synthesize triglycerides. Triglycerides, in turn, can perform or can be involved, I should say, different reactions, including hydrolysis under acidic conditions, which are going to produce glycerol and fatty acids, meaning an alcohol and a carboxylic acid. Triglycerides can also be hydrolyzed under basic conditions. And when that happens, the products of the reaction are going to be glycerol and carboxylic acid salts. And this fatty acid salts or carboxylic acid salts is what we know as a soap. Triglycerides can also undergo hydrogenation. And when a triglyceride is hydrogenated, partially or fully, let's understand that the product of that reaction is going to be a more saturated triglyceride.